Hello there, and welcome back to Bedtime Science. I'm really glad you're here tonight. Tonight, we are going to follow one of the quietest and most complicated performances the human body ever gives, the work of healing. Healing is easy to overlook when it succeeds. A small cut closes. A bruise changes color and disappears. A strained muscle becomes useful again. A fever passes. A broken bone, held still for long enough, slowly becomes one piece. The evidence of damage fades, and ordinary life returns so gradually that the work behind it can seem almost automatic. But nothing about recovery is simple. The body does not keep a tiny copy of itself hidden away, ready to replace every damaged part. It must detect what has changed, stop what is leaking, remove what can no longer be saved, defend against infection, build temporary structures, grow new vessels, produce new tissue, and then reshape that tissue for weeks, months, or even years. It must do all of this while keeping the rest of the body alive and functioning. Healing is less like pressing a reset button and more like rebuilding a city while people are still living in it. Tonight, we will begin with the smallest imaginable injury, a narrow break in the skin. From there, we will follow the messages that travel through blood and tissue. We will meet platelets that become architects of a temporary seal, immune cells that arrive first as defenders and later as organizers, fibroblasts that pull a wound inward, and new capillaries that grow toward tissue in need of oxygen. Then the story will widen. We will watch a broken bone build a bridge across a gap. We will see muscle awaken its reserve cells after strain. We will follow a damaged peripheral nerve as it clears a path and attempts to grow again. We will visit the liver, an organ whose ability to restore its mass has fascinated physicians for centuries, and the brain, where recovery often depends less on replacing lost cells than on teaching surviving networks to work in new ways. We will also look at what happens during illness, why the immune system can make the body sleepy and still, why inflammation must begin and then know when to end, and why sleep is not an empty pause in the healing process. Sleep changes hormones, immune signaling, memory, pain, metabolism, and the way the brain handles the day's activity. Rest does not perform every repair by itself, but it creates conditions in which many forms of recovery can proceed more effectively. And eventually, we will arrive at the frontier of regenerative medicine, where researchers study salamanders that rebuild limbs, zebrafish that repair injured hearts, and human tissues grown in carefully designed environments. The goal is not to turn biology into magic. It is to understand why some tissues repair with scars, why others regenerate more faithfully, and whether those choices can be influenced. For now, imagine only the skin. The skin looks still, but it is one of the busiest organs in the body. Its outer layers are continually renewed. Cells born deeper in the epidermis gradually move upward, change their structure, fill with protective proteins, and finally become part of the tough outer barrier before being shed. Oils, sweat, immune cells, nerves, blood vessels, hair follicles, and communities of microorganisms all share this surface. The skin is not simply wrapping. It is a border, a sensor, a temperature regulator, a water barrier, and an immune environment. It separates the controlled chemistry inside the body from a world filled with dryness, friction, sunlight, microbes, and sudden sharp edges. Now imagine that a thin piece of paper catches a fingertip. The cut is small, but from the body's point of view, several things have happened at once. Cells have been torn. Tiny blood vessels have opened. The protective barrier has been breached. Materials normally hidden inside cells have spilled into the surrounding space. Microbes from the surface may have gained a path inward. Nerve endings have been disturbed, producing the quick, bright message of pain. The first response begins within seconds. The damaged blood vessel narrows. Smooth muscle in the vessel wall contracts, reducing the flow of blood. This vasoconstriction is brief, but it buys time. At the exposed surface beneath the vessel lining, proteins that blood normally does not encounter become visible. Among them is collagen, a structural protein familiar from skin, tendons, bone, and many other tissues. Platelets passing in the bloodstream recognize that something is wrong. A platelet is not a complete cell in the usual sense. It is a small fragment released from enormous bone marrow cells called megakaryocytes. In quiet circulation, platelets drift in a smooth, inactive form. At an injury, they change. They adhere to exposed structures, assisted by proteins including von Willebrand factor, which acts as a molecular bridge between platelet and damaged vessel wall. Once activated, the platelets become sticky. They spread, extend projections, release chemical signals, and recruit more platelets from the passing blood. What begins as a few fragments attaching to an injured surface becomes a growing plug. This early plug is fast, but fragile. It needs reinforcement. At the same time, a chain of reactions begins among proteins already dissolved in the blood. This is the coagulation cascade. The name suggests a simple row of falling dominoes, but the real network contains several interacting paths, amplifications, checks, and surfaces. Its purpose is to generate an enzyme called thrombin in the right place and at the right time. Thrombin changes soluble fibrinogen into fibrin. Fibrin molecules join into long threads that spread through the platelet plug like ropes through wet concrete. The threads are cross-linked into a stronger mesh. Red blood cells and other components become caught within it. The temporary seal becomes a clot. A clot is often imagined only as something that stops bleeding. It is more than a cork. It is the first provisional landscape of repair. Within the fibrin network are platelets carrying stored signals. Around the clot are fragments of damaged tissue, proteins from plasma, and chemical messages released by stressed cells. Together, they form a temporary matrix where immune cells can arrive and where later repair cells can attach, migrate, and communicate. The clot is designed to be temporary. If it were permanent, it would block rebuilding. So even while the body forms fibrin, it also prepares a system that will eventually remove it. Healing begins with construction and demolition planned at the same time. This balancing act is essential throughout the body. Blood must clot quickly at an injury, but remain fluid everywhere else. A clot that forms too slowly can allow dangerous bleeding. A clot that forms where no vessel is damaged can obstruct blood flow. The same chemistry that saves blood at a wound must be carefully restricted by the vessel lining, circulating inhibitors, and the flow of blood itself. At the paper cut, the scale is tiny. The balance is still extraordinary. The quick pain of the injury may fade into throbbing or tenderness. Pain is not simply a report of damage. It is a protective instruction. Sensory nerves respond to mechanical disturbance, heat, acidity, and inflammatory chemicals. Their signals travel toward the spinal cord and brain, where the experience of pain is constructed from the incoming message, attention, memory, emotion, and context. The sore finger is treated differently because it hurts. Grip changes. Pressure is avoided. The injured tissue is granted a small measure of protection while early repair begins. Pain can therefore serve healing, but pain and damage are not identical. Severe pain can occur without severe tissue injury, and serious injury can sometimes produce surprisingly little pain at first. As recovery continues, the nervous system may increase or decrease sensitivity. In some conditions, protective pain outlasts its original purpose and becomes a problem of its own. The relationship between healing and pain is a conversation, not a simple meter. For the moment, the pain has done something useful. It has made the injury impossible to ignore. Beneath the skin, damaged cells release alarm signals, sometimes called damage-associated molecular patterns. These are ordinary molecules in unusual places. A molecule that belongs inside a healthy cell may become a warning when found outside it. Nearby immune cells detect the change through receptors shaped by evolution to recognize both microbes and signs of tissue damage. Mast cells release mediators. Blood vessel walls become more permeable. Local vessels widen after the first brief constriction. Warm blood reaches the area. Fluid and proteins move from the circulation into the tissue. The finger may become red, warm, slightly swollen, and tender. These are the classic signs of inflammation. The word inflammation is often used as though it names an enemy. In long-lasting disease, excessive or misdirected inflammation can damage tissue. But acute inflammation after injury is not a mistake. It is the opening phase of repair. The injured site must be cleaned. Microbes must be controlled. Dead cells and broken matrix must be removed. Signals must be sent to the cells that will rebuild. Without a properly timed inflammatory response, healing becomes weak or disorganized. The challenge is not simply to suppress inflammation. It is to begin it, shape it, and then resolve it. Neutrophils are among the first immune cells to arrive in large numbers. They circulate in the blood, waiting for signals that tell them where vessel walls have become activated. Near an injury, they roll along the inner vessel surface, attach more firmly, squeeze between endothelial cells, and move through tissue toward chemical gradients. Their journey is guided by concentration. Molecules are more abundant near the damaged site, and the neutrophils follow that rising trail, changing direction through spaces far smaller than their resting shape. Neutrophils are rapid defenders. They engulf microbes and debris. They release enzymes and antimicrobial substances. In some circumstances, they cast out networks of DNA and proteins that can trap microorganisms. Their methods are powerful, and because power can harm nearby healthy tissue, their activity must be controlled. Most neutrophils do not remain for long. They perform their work, undergo programmed cell death, and are removed by other immune cells. The arrival and disappearance of neutrophils is part of the wound's changing rhythm. Macrophages soon become central. Some macrophages already live in tissues. Others develop from monocytes recruited from the blood. Their name means large eater, and they do engulf dead cells, microbes, and fragments. But describing a macrophage only as a cleaner misses its most remarkable role. Macrophages interpret the condition of the tissue and help direct what happens next. Early in inflammation, many macrophages produce signals that support defense and amplify the response. They help recruit other cells and remove dangerous material. Later, after engulfing dying neutrophils and sensing changes in their environment, macrophages can shift their behavior. They release signals that encourage blood vessel growth, fibroblast activity, matrix production, and the resolution of inflammation. Popular explanations sometimes divide macrophages into two simple types: inflammatory and healing. The real biology is more flexible. Macrophages occupy a wide range of states shaped by tissue, time, metabolism, microbes, and neighboring cells. They do not flip one universal switch. They change programs as the wound changes. This transition is one of the most important moments in healing. A defensive wound cannot remain defensive forever. Molecules that recruit immune cells must decline. Dead cells must be removed without spilling more alarm signals. Anti-inflammatory and pro-resolving pathways must become active. New tissue must be allowed to grow. Resolution is not the same as inflammation simply running out of fuel. It is an active process. Specialized lipid mediators, changes in cytokines, removal of inflammatory cells, and shifts in macrophage behavior help bring the response toward repair. The body has begun by sealing the breach and calling an emergency crew. Now it must decide when the emergency phase is over. That decision is rarely made by one cell or one molecule. It emerges from many local conversations. And while those conversations continue beneath the small cut, another group of cells begins moving across the wound from the edges. They are keratinocytes, the main cells of the epidermis, and they are preparing to close the surface again. Keratinocytes normally sit in an orderly layered community. Near the wound edge, they change their attachments, flatten, and begin to migrate. They move over the temporary matrix beneath them, advancing from both sides like a living sheet. They do not wait for the gap to be filled completely from below. Re-epithelialization begins early. Cells crawl beneath a dry scab when one is present, using the moist tissue below as their path. This is one reason the scab itself is not the new skin. It is a temporary roof while the real surface closes underneath. The moving keratinocytes communicate with immune cells, fibroblasts, and neighboring tissue. They release signals, respond to growth factors, and change which genes they use. Cells behind the leading edge divide to supply more cells. When the advancing sheets meet, migration slows and the epidermis begins rebuilding its layers. Below them, the wound is becoming filled with granulation tissue. The name can sound rough, but healthy granulation tissue is often soft, pink or red, and richly supplied with tiny blood vessels. It contains fibroblasts, immune cells, extracellular matrix, and new capillaries growing into the damaged space. Angiogenesis, the formation of new blood vessels from existing ones, is essential because rebuilding is expensive. Dividing cells need oxygen and nutrients. Waste products must be carried away. A wound that has lost its circulation cannot simply wait for blood to return on its own. Endothelial cells lining nearby vessels respond to signals, such as vascular endothelial growth factor. Some become leading tip cells, extending delicate projections through the matrix. Others follow, forming the walls of new capillary sprouts. The sprouts branch, connect, and gradually establish loops through which blood can flow. For a time, these new vessels are fragile and leaky. That is part of why fresh granulation tissue may bleed easily. Over time, vessels mature, recruit supporting cells, and some of the temporary network is removed when it is no longer needed. The wound grows its own roads before the permanent buildings are complete. Fibroblasts are among the principal builders. They migrate into the provisional matrix and produce collagen, fibronectin, proteoglycans, and other components of extracellular matrix. This matrix is not inert packing. It gives cells places to attach, influences their shape, stores signals, and helps determine how forces move through the tissue. Early collagen is laid down quickly and without the elegant organization of uninjured skin. Speed matters more than perfection. A weak bridge built today can be strengthened tomorrow. Some fibroblasts become myofibroblasts. These cells share features with smooth muscle and can contract. They attach to the matrix and pull, drawing the wound edges closer together. Wound contraction reduces the area that must be filled, especially in larger open wounds. The pulling is coordinated through networks of actin and molecular attachments. Each cell exerts only a small force. Across thousands of cells connected to the matrix, the wound visibly narrows. This is tissue behaving mechanically. Cells sense stiffness, tension, and stretch. They respond by changing gene activity, movement, and the material they produce. Healing is chemical, but it is also physical. A wound is a place where forces are measured continuously. As closure continues, the clot is dismantled. Fibrin is broken down by enzymes in the fibrinolytic system. Temporary matrix is replaced by stronger matrix. Cell populations that were essential early begin to decline. Capillaries that are no longer required regress. The wound becomes less red and less cellular. Then begins the longest phase, remodeling. Collagen fibers are cut, reassembled, and aligned according to mechanical stress. Type 3 collagen, common in early repair, is increasingly replaced by stronger type 1 collagen. Crosslinks form. The scar becomes more resistant to tension. But repaired skin does not usually return to exactly the same structure it had before injury. Hair follicles, sweat glands, pigment patterns, nerves, and the basket-like arrangement of collagen may not be fully restored. The scar is a solution, not a perfect rewind. Even after maturation, scar tissue usually does not regain the full tensile strength of uninjured skin. It becomes strong enough for ordinary life, but the architecture remains different. This difference is not necessarily failure. Evolution has often favored rapid closure over flawless reconstruction. An open wound threatens blood loss and infection. A scar may be less elegant, less flexible, and less complete, but it seals the body quickly. The scar is evidence that survival came first. Some scars become raised or excessive. Hypertrophic scars remain within the original wound boundaries, while keloids can extend beyond them. Their biology involves prolonged fibroblast activity, collagen production, tension, genetic influences, and altered signaling. Why one person forms a fine pale line and another forms a thick growing scar is still not explained by one simple cause. Scars also change over time. A new scar may be red, firm, itchy, or sensitive. Months later, it may flatten, soften, and fade as vessels regress and matrix is remodeled. Nerves recovering in the area may produce tingling or strange sensations. The visible surface is only the final expression of slower changes below. Itching is common during healing, though its precise causes vary. Dryness, inflammatory mediators, nerve activity, and skin tension may all contribute. The sensation is a reminder that recovery has its own sensory language. Healing tissue can feel tight, warm, numb, electric, or simply unfamiliar. The body does not rebuild silently. It sends reports. For most of human history, people could see wounds close without knowing the cellular events beneath them. They knew that some injuries healed and others became swollen, foul, feverish, and deadly. They did not know that invisible microorganisms could enter through broken skin or that hands, instruments, and dressings could carry them. The history of healing changed when infection became understandable as biology rather than fate. In the 19th century, surgery was becoming faster and technically more ambitious, but postoperative infection remained common. A surgeon might complete an operation successfully and still lose the patient days later to sepsis or gangrene. Hospital wards could carry the smell of infected wounds. Pus was sometimes treated as an expected part of healing. Ignaz Semmelweis noticed a devastating pattern in maternity clinics in Vienna. Women attended by physicians and medical students died of childbed fever far more often than women attended by midwives. The students moved from autopsy rooms to deliveries, carrying material on hands that appeared clean to the eye. Semmelweis required hand cleansing with a chlorinated solution, and mortality fell dramatically. Yet his explanation came before germ theory was widely accepted. The suggestion that physicians themselves transmitted death was resisted, and his insight did not receive the recognition it deserved during his life. Later, Louis Pasteur's work helped establish that microorganisms did not arise spontaneously from decay. Joseph Lister applied germ theory to surgery. Using antiseptic methods, he attempted to reduce contamination of wounds, instruments, dressings, and the surgical environment. The early methods were imperfect and sometimes harsh, but the principle transformed medicine. A wound was not sealed from danger merely because the bleeding had stopped. Invisible life could enter, multiply, and redirect the entire healing response. Modern infection control grew far beyond carbolic spray. Sterilization, clean technique, antibiotics, vaccination, careful wound care, and understanding of microbial ecology have all changed the outcome of injury. Yet the basic insight remains. Healing is a collaboration between tissue repair and control of microbes. The immune system must discriminate. It should respond strongly enough to contain infection, but not so destructively that it destroys the tissue it is meant to protect. Many of the same molecules that kill microbes can damage host cells. A prolonged battle can turn repair into chronic inflammation. In an acute wound, the sequence moves forward. Clot, inflammation, new tissue, remodeling. In a chronic wound, the sequence may stall. Blood flow may be inadequate. Repeated pressure may injure the same area again. High glucose can alter immune function, vessels, and nerves. Bacteria may form biofilms, communities protected by a matrix that makes them harder to clear. Edges may fail to migrate. Fibroblasts may become less responsive. Inflammation may remain active without completing the transition to repair. The wound is busy, but not progressing. This distinction matters because healing is not measured by how much activity exists. It is measured by whether activity changes at the right time. An orchestra that holds one loud note forever is not performing the full composition. The same principle appears in inflammation throughout the body. Acute inflammation can defend and repair. Chronic inflammation can alter vessels, joints, lungs, fat, liver, and nervous tissue. The difference is not merely intensity. It is duration, location, cause, and the failure of resolution. Recovery depends on timing. There is a time for platelets to stick and a time for the clot to dissolve. A time for neutrophils to attack and a time for them to disappear. A time for fibroblasts to build and a time for them to reduce their work. A time for protection and a time for movement. The body's intelligence is often temporal. The same signal can help at one stage and harm at another. This is especially clear in bone, where healing must solve an engineering problem more demanding than closing skin. A broken bone must restore continuity, alignment, blood supply, and the ability to carry force. It must build something rigid without becoming rigid too early. Imagine a crack through a long bone. At the instant of fracture, blood vessels within the bone and surrounding tissue tear. A hematoma forms around the break. Cells die near the damaged edges because their blood supply is lost. Pain signals protect the limb. Muscles may spasm. Swelling begins. The first structure joining the broken ends is not bone. It is blood. The fracture hematoma is more than pooled blood. It is a biochemical signal that a structure designed to bear weight has failed. Platelets activate. Fibrin forms. Immune cells arrive. Oxygen levels fall within parts of the damaged region. Cells respond to the unusual combination of inflammation, low oxygen, disrupted matrix, and mechanical instability. Bone is sometimes pictured as a dead mineral frame, but living bone is crowded with cells and vessels. Osteocytes sit within tiny spaces in the mineralized matrix and communicate through microscopic channels. Osteoblasts build bone. Osteoclasts resorb it. Marrow contains blood-forming cells and stromal populations. The outer periosteum and inner endosteum hold cells capable of contributing to repair. Soon after fracture, inflammatory signals recruit immune and progenitor cells. Macrophages clear damaged material and help coordinate the transition toward reconstruction. New vessels begin growing into the area. Oxygen returns unevenly. The mechanical environment around the break begins influencing what kind of tissue forms. If the fracture is held in a suitable position but retains some controlled movement, the body often uses secondary bone healing. A soft callus appears first. This is a bridge made partly from fibrous tissue and cartilage. It stabilizes the ends without pretending that the final structure already exists. Cartilage is useful here because it can form and survive in a relatively low oxygen environment. Cells derived from local progenitors become chondrocyte and produce a flexible matrix. The callus expands around and between the broken ends, reducing movement. Then the soft callus becomes a hard callus. Blood vessels invade. Cartilage is gradually removed and replaced by woven bone through a process related to the way much of the skeleton formed during development. Osteoblasts lay down mineralized matrix. The bridge becomes rigid enough to carry increasing force. Woven bone is built quickly. Its collagen fibers are arranged irregularly, giving rapid strength, but not the refined organization of mature bone. Later, remodeling replaces it with lamellar bone whose structure is aligned more efficiently with stress. Osteoclasts remove material. Osteoblasts fill new surfaces. The bulky callus is sculpted. The marrow cavity may reopen. The outer shape approaches what it was before the fracture. This can continue for months or years after the bone first appears healed on an image. Bone does something skin usually cannot. Under good conditions, it can restore tissue so completely that little or no scar remains at the fracture site. The repaired region can become normal living bone capable of continued remodeling. This remarkable result depends on biology and mechanics working together. Too much movement can prevent a stable bridge from forming. Too little useful load over long periods can reduce bone and muscle. Absolute stability can support a different form of healing in which bone crosses the fracture through direct remodeling with little callus. Surgeons choose fixation methods according to fracture pattern, location, blood supply, tissue damage, and the mechanical goal. A plate, nail, screw, cast, or external frame does not heal the bone. It changes the environment so that cells can heal it. This is an important distinction throughout medicine. Many treatments do not perform recovery in place of the body. They protect, align, supply, remove obstacles, or create conditions in which biology can proceed. Bone responds to loading even when uninjured. Osteocytes sense strain and fluid movement within their tiny channels. Signals influence the balance between formation and resorption. Areas exposed to repeated useful force can be strengthened. Areas kept without load can lose mass. The skeleton is not a building completed in childhood. It is a structure under continuous renovation. Old bone accumulates microscopic damage. Remodeling units remove small regions and replace them. Calcium and phosphate are exchanged with the rest of the body. Hormones, nutrition, activity, age, and disease alter the balance. A skeleton can look still while millions of microscopic construction sites are active inside it. The same mechanical sensitivity makes rehabilitation after fracture a careful progression. Early protection prevents disruption. Later movement and loading provide information that helps tissues organize. The exact timing depends on the injury and treatment. Biology must be challenged enough to adapt, but not overwhelmed before it can respond. Healing is neither endless rest nor constant effort. It is a sequence of changing needs. Muscle offers another version of that sequence. A skeletal muscle is made from long fibers containing many nuclei. Inside each fiber, repeating units of actin and myosin slide past one another to produce force. Motor nerves activate groups of fibers. Blood vessels deliver oxygen and fuel. Connective tissue transmits force toward tendons and bone. During unfamiliar or intense exercise, especially lengthening contractions in which a muscle produces force while being stretched, small areas of the muscle and surrounding structures can be disturbed. This is not always a dramatic tear. It may be microscopic disruption, altered membranes, stressed proteins, and an inflammatory response. The soreness that appears a day or two later is called delayed onset muscle soreness. It is not caused primarily by lactic acid remaining in the muscle. Lactate is cleared far sooner. The delayed discomfort reflects a changing response involving local damage, inflammation, fluid shifts, and sensitized nerve endings. The delay itself is revealing. The moment of strain and the experience of soreness are separated because biology takes time to build the response. Muscle fibers are large and specialized, but they have a reserve population nearby, satellite cells. These cells lie between the muscle fiber membrane and its surrounding basal lamina. In resting muscle, they are quiet. After injury or sufficient growth signals, some become activated. They divide. Some descendants contribute new nuclei to existing fibers or help rebuild damaged regions. Others return to a reserve state, preserving the population for future repair. Satellite cells are not working alone. Neutrophils and macrophages arrive. Fibroadipogenic progenitors help organize the connective environment but must be controlled so that repair does not become excessive fibrosis or fat deposition. Blood vessels respond. Motor nerves matter. Mechanical loading changes signals. A regenerating muscle is an ecosystem. Early inflammatory macrophages help clear damaged material. Later macrophage states support cell growth and differentiation. The timing resembles wound healing because many tissues use related cellular languages. Yet the outcome is adapted to muscle. New contractile structure must align with the old fiber and reconnect to force transmitting networks. Protein synthesis increases after suitable exercise and nutrition, but muscle recovery is not simply filling a torn space with protein. Existing proteins are removed or repaired. Ribosomes build new proteins from amino acids. Nuclei regulate local regions of the enormous fiber. Mitochondria adjust in number and function. Capillaries can change with training. Neural recruitment becomes more efficient. Recovery from exercise and adaptation to exercise overlap. The body does not merely restore what existed before. It may prepare for a similar demand in the future. Muscle fibers can enlarge. Enzymes change. Tendons adapt more slowly. The nervous system learns the movement. The same challenge that temporarily reduces performance can, after recovery, increase capacity. This is the principle of adaptation, but adaptation is not guaranteed by damage alone. Too little stimulus produces little change. An appropriate challenge followed by sufficient recovery can produce improvement. Repeated overwhelming stress can outpace repair and lead to injury, fatigue, or loss of function. The useful signal lies within a range. Tendons make the contrast especially clear. A tendon connects muscle to bone and transmits enormous forces through collagen fibers arranged mainly along the direction of pull. Tendon cells, called tenocytes, maintain the matrix. The tissue is strong but relatively less vascular than many muscles, and its remodeling is slow. After tendon injury, inflammation, cell proliferation, collagen production, and remodeling occur, but the regenerated structure may remain less organized than the original. Early collagen is thin and disordered. Over time, fibers align with loading, cross-links change, and mechanical strength improves. Recovery can continue long after pain has eased. Because tendon adapts slowly, the muscle attached to it may feel capable before the tendon has fully remodeled. The strongest part of the movement system can therefore be ready before the slower part. Ligaments share some of these challenges. They connect bone to bone and guide joints. Their healing varies by location, blood supply, mechanical environment, and whether the torn ends remain near one another. Some repair with scar tissue. Some injuries require reconstruction because spontaneous healing cannot restore useful stability. Cartilage is more limited still. Articular cartilage covers the ends of bones inside joints, creating a smooth, low-friction surface. It has no direct blood supply. Chondrocytes live within a dense matrix and receive nutrients through diffusion from joint fluid and nearby tissues. When cartilage is damaged, especially through its full thickness, its ability to restore the original architecture is limited. The absence of vessels protects cartilage from some kinds of inflammation, but also deprives it of easy access to repair cells and signals. A tissue beautifully adapted for low-friction movement is poorly equipped for rapid rebuilding. Different tissues heal differently because they are different solutions to different problems. Skin must seal quickly. Bone must carry force. Muscle must contract. Tendon must transmit tension. Cartilage must glide with almost no friction. Nerve must conduct information over distance. The architecture that makes each tissue excellent at its everyday job also shapes the way it can recover when damaged. The nervous system presents one of the most striking divisions. A peripheral nerve in the arm or leg has some capacity to regrow. A damaged pathway in the brain or spinal cord faces far greater obstacles. To understand why, imagine a single nerve fiber running from a motor neuron near the spinal cord toward a muscle in the foot. The axon may extend for nearly a meter, yet it belongs to one cell. Proteins, membranes, organelles, and signals must travel along that extraordinary distance. If the nerve is cut, the part of the axon separated from the cell body cannot survive indefinitely. It undergoes Wallerian degeneration. The distal segment breaks down. Myelin unravels. Macrophages and Schwann cells clear debris. At first, this sounds like further destruction. In fact, clearing the old pathway is part of making regeneration possible. Schwann cells, which normally wrap peripheral axons and form myelin, change their identity after injury. They reduce their myelinating program, multiply, clear debris, release growth-supporting signals, and align within the old nerve tubes. These columns can guide new axonal sprouts from the surviving proximal stump. The nerve prepares a road through the ruins. From the cut end, the injured axon sends out several sprouts. Many will fail. One may enter the correct Schwann cell channel and continue toward its old target. The growth cone at its tip explores the environment, responding to chemical and mechanical cues. New membrane and internal scaffolding are added as the axon lengthens. Peripheral axons are often described as growing roughly 1 to a few millimeters per day, though the real rate varies with nerve, distance, age, injury, and conditions. At that speed, a pathway to a distant hand or foot may require months. The muscle and sensory receptors waiting at the far end cannot remain unchanged forever. Time matters. If the axon reaches its original target, function may gradually return. Myelin is rebuilt, though often thinner and shorter between nodes than before. The nervous system must learn to interpret the returning signals. Muscles weakened by disconnection must recover. Sensory maps may feel distorted. Pins and needles, numbness, hypersensitivity, and awkward movement can accompany the rebuilding. Recovery is therefore not finished when the axon arrives. The cable, insulation, target, and brain must all become coordinated again. The central nervous system is less permissive. After injury in the brain or spinal cord, myelin-associated molecules can inhibit axon growth. A dense glial scar forms. Inflammation and cell death create a complex environment. Long axons may not regenerate across the damaged region in a useful way. Yet the brain possesses another form of recovery: plasticity. Neural plasticity is the ability of circuits to change their strength, structure, and function. It supports learning throughout life, and after injury, it can help surviving networks take on new roles. Recovery after a stroke, for example, may involve tissue around the injured area, corresponding regions in the opposite hemisphere, and broader networks adjusting their connections. A stroke does not simply erase one skill from one location. It interrupts blood flow to a region and affects networks that depend on it. Some cells die quickly. Others are stunned but potentially salvageable. Swelling, inflammation, and altered metabolism influence the early period. Later, the challenge becomes reorganization. A person may practice reaching toward a cup again and again. Each attempt is a movement, but also an experiment performed by the nervous system. Which muscles activate? Which sensory signals return? Which pathway can be strengthened? The visible action may be small while the network-level work is extensive. Rehabilitation uses repetition, attention, feedback, task difficulty, and meaningful goals to guide plasticity. The brain changes in response to experience, whether that change is helpful or not. Avoided movements can become more difficult. Compensatory habits can become strong. Carefully designed practice tries to make useful patterns more likely. The phrase "the brain rewires itself" is convenient, but it can sound too simple. New synapses may form. Existing synapses may strengthen or weaken. Dendritic branches may change. Myelin can adapt. Networks can alter timing and recruitment. Some functions shift. Others remain limited by the location and extent of injury. Plasticity creates possibility, not a promise of complete restoration. Recovery also has a psychological landscape. Frustration changes attention. Fear of falling changes movement. Confidence influences willingness to attempt a difficult task. Fatigue limits practice. Family, therapists, environment, and access to care affect what the nervous system experiences repeatedly enough to learn. A healing brain is not isolated from a healing life. Memory shows how recovery and reconstruction can blur. Every time a memory is recalled, it can become temporarily changeable before being stored again, a process called reconsolidation. This does not mean memories can be edited at will like documents. It means remembering is active. The brain rebuilds a present version from distributed traces. After trauma, pain, or illness, the nervous system may learn associations that remain protective after danger has passed. A movement linked with injury may continue to feel threatening. A smell linked with a hospital may provoke distress. Recovery can involve new learning that places the old signal in a safer context. The body repairs tissue, while the brain repairs predictions. Pain illustrates this predictive work. Nociceptors detect potentially damaging conditions, but pain itself is produced by the nervous system as a protective experience. In acute injury, pain usually tracks the need for protection reasonably well. In persistent pain, the alarm system may remain sensitized. The spinal cord and brain can amplify signals. Harmless touch may hurt. Movement may feel dangerous even when tissue has largely healed. This does not make the pain imaginary. It makes it biological at another level. Recovery from persistent pain often requires more than waiting for tissue. It may involve calming sensitivity, restoring movement, improving sleep, addressing fear, and changing the nervous system's interpretation of threat. The exact approach depends on the condition, and no single explanation fits every person. The nervous system is both witness to injury and participant in recovery. Some tissues replace their cells continually even without obvious damage. The lining of the intestine is one of the fastest renewing surfaces in the body. It faces digestive enzymes, mechanical stress, food, microbes, and chemicals while maintaining a barrier only 1 cell thick in many regions. Deep pockets called crypts contain stem and progenitor cells. Their descendants divide, move upward, specialize, perform their functions, and are shed. The surface is renewed in a matter of days. This speed is possible because the architecture preserves a protected source of new cells. Signals from neighboring cells and matrix create a stem cell niche, a local environment that balances self-renewal with differentiation. Too little division and the barrier fails. Too much uncontrolled division and cancer risk rises. Renewal is powerful because it is regulated. Blood is also renewed continuously. Red blood cells circulate for roughly 4 months before being removed and recycled. Bone marrow produces replacements from hematopoietic stem cells. White blood cells have lifespans ranging from hours to years depending on type and state. Platelets survive for days before new fragments take their place. The blood that sealed the paper cut is itself a moving population under constant replacement. Skin, intestine, and blood can renew rapidly because their everyday function includes turnover. Other tissues preserve long-lived cells. Heart muscle cells in adults have only limited capacity to divide. Many neurons are expected to last for decades. The strategy is maintenance rather than frequent replacement. This creates a trade-off. A long-lived cell can preserve specialized structure and information, but losing it may be difficult to reverse. The liver occupies a remarkable middle ground. Ancient mythology already associated the liver with renewal. In the story of Prometheus, an eagle consumes his liver, and the organ grows again before the next day's punishment. The myth is cruel, but its choice of organ is strangely appropriate. The human liver can restore its mass after a substantial portion is removed, provided the remaining tissue is healthy enough and blood supply is maintained. It does not usually regrow the missing lobe in exactly the same shape. Instead, the remaining lobes enlarge until functional mass is restored. This is compensatory growth rather than a perfect anatomical copy. Most liver cells called hepatocytes are normally quiet, performing metabolism, protein synthesis, detoxification, bile production, and countless chemical tasks. After loss of tissue, signals from blood flow, cytokines, growth factors, bile acids, and neighboring cells prompt many hepatocytes to re-enter the cell cycle. The organ rebuilds while continuing to regulate the chemistry of the body. Kupffer cells, the liver's resident macrophages, help initiate signaling. Endothelial cells, stellate cells, immune cells, and bile duct cells participate. The extracellular matrix changes. Blood vessels adapt. Later, growth must stop when sufficient mass returns. Again, the mystery is not only how cells divide. It is how they know when to stop. Liver regeneration has limits. Chronic injury can lead to fibrosis, in which scar tissue accumulates and distorts the architecture. Repeated cycles of cell death and inflammation may produce cirrhosis. In that setting, regenerative signals continue, but the tissue environment becomes less supportive and more disorganized. A strong regenerative capacity does not make an organ invulnerable. The contrast between liver and heart has driven decades of research. Why can one adult organ restore mass, while another often heals by scar? After a heart attack, blood supply to part of the heart muscle is blocked. Cardiomyocytes die. Immune cells clear debris. Fibroblasts produce collagen. A scar forms that prevents the ventricular wall from rupturing, but the scar does not contract like heart muscle. This is a life-saving repair that reduces pumping capacity. The adult human heart replaces cardiomyocytes only slowly. It cannot generally rebuild a large lost region with new beating muscle. Researchers study developmental signals, metabolism, cell cycle control, immune responses, extracellular matrix, and electrical integration in search of ways to improve regeneration. Other animals reveal what is biologically possible. Adult zebrafish can regenerate a substantial portion of injured heart tissue. Existing cardiomyocytes near the injury alter their state, divide, and contribute new muscle. Blood vessels and supportive cells respond. Temporary scar-like tissue is resolved more fully than in the adult mammalian heart. Newborn mice retain a brief period of stronger heart regeneration that fades soon after birth. The loss suggests that mammalian cardiomyocytes do not lack every regenerative program. Some programs become restricted as the heart matures, metabolism changes, oxygen exposure rises, and cells specialize. Scientists are not simply trying to copy a fish. They are asking which switches differ, which trade-offs protect adult mammals, and whether repair can be improved without causing arrhythmia, weak tissue, or uncontrolled cell growth. Every regenerative power carries risks. A cell asked to divide must copy DNA. A cell asked to become less specialized may lose stable identity. Growth signals can support repair, but cancer also exploits growth signals. A tissue that regenerates rapidly must still preserve shape, polarity, blood supply, and function. Healing is controlled growth under pressure. Cancer can be viewed, in part, as growth escaping the rules that make repair temporary. Tumors recruit vessels, remodel matrix, influence immune cells, resist death, and maintain proliferative signals. Many of the pathways are not foreign inventions. They are ordinary biological tools used at the wrong time, in the wrong place, without a proper stop. The same molecules can participate in development, wound healing, and disease. This is why regenerative medicine moves carefully. The goal is not simply more growth. It is the right cells, in the right arrangement, for the right duration, connected to nerves and vessels, and able to stop when the work is complete. The body's most impressive repairs are not the fastest. They are the ones that end well. Healing after infection begins with a different kind of alarm. A virus enters a cell. A bacterium crosses a barrier. A parasite occupies tissue. The immune system must recognize something that may be microscopic, changing, and hidden among the body's own molecules. Innate immunity responds first with patterns. Cells carry receptors that recognize features common to broad groups of microbes. Unusual forms of RNA, bacterial cell wall components, or molecular arrangements rarely exposed in healthy tissue. An infected cell can also detect its own distress and release interferons, signals that warn neighboring cells and change their behavior. The response spreads beyond the original site. Cytokines enter circulation. Bone marrow adjusts the production and release of immune cells. The liver changes which proteins it sends into blood. The brain receives messages through nerves, blood vessels, and specialized interfaces. Then the person begins to feel ill. Tiredness, loss of appetite, reduced interest in ordinary activity, increased sleepiness, body aches, and a desire to withdraw are grouped under the term sickness behavior. These experiences are not merely the passive result of damage. They are partly organized responses to immune signaling. The immune system changes motivation. This makes evolutionary sense. Activity is expensive. Searching for food, competing, exploring, and maintaining social behavior require energy. During infection, energy is being redirected toward fever, immune cell activity, protein production, and tissue maintenance. Reduced activity may also lower exposure to additional hazards and, in social species, sometimes reduce transmission. Sickness behavior is useful when it is temporary and proportional. Like inflammation, it becomes harmful when prolonged or disconnected from an acute need. Fever is another controlled shift. Body temperature is regulated around a changing set point. During many infections, molecules including prostaglandin E2 influence the hypothalamus, raising the target. The existing body temperature now feels too low. Blood vessels near the skin constrict. Muscles may shiver. The person seeks warmth even as the measured temperature rises. Later, when the set point returns downward, the same temperature feels too high. Vessels widen. Sweating increases. Heat leaves the body. Fever is not simply accidental overheating. It is a regulated state that can influence microbial growth and immune activity. Yet high temperature also increases metabolic demand and can be dangerous in particular situations, especially in vulnerable people. Biology rarely offers one universal meaning independent of context. The body's response to infection can feel unpleasant precisely because it is coordinated. Aches arise partly from inflammatory mediators, changing pain sensitivity. Fatigue reflects signals reaching the brain, altered metabolism, and the cost of immune activity. Appetite can decline while the liver and immune system redistribute nutrients. Sleep becomes fragmented in some infections and increased in others. The subjective experience is the surface of a whole body program. Adaptive immunity adds specificity and memory. B cells can produce antibodies whose shapes bind particular molecular targets. T cells recognize fragments presented by other cells and can help coordinate responses or kill infected cells. During a first encounter, rare lymphocytes with useful receptors are activated and multiply. Some become short-lived effectors. Others remain as memory cells. The next encounter can be faster because the body no longer begins with only a few matching cells. Memory is not perfect and not permanent for every pathogen. Microbes evolve. Antibody levels change. Different tissues require different forms of protection. Still, immunological memory is one of healing's most important forms of learning. The body can recover from an infection and retain information about it. Vaccination uses this capacity without requiring the full disease. A vaccine presents an antigen, genetic instruction, or harmless version of a pathogen in a way that trains immune memory. The response may cause temporary soreness, fatigue, or fever because the immune system is practicing parts of the same communication used during infection. Recovery can therefore begin before exposure, through preparation. After a pathogen is controlled, the immune response must contract. Most of the expanded effector cells die. Antibody production changes. Macrophages clear debris. Tissue-resident cells restore barriers. Regulatory pathways reduce activation. The end of infection is not the instant the last microbe disappears. It is a long unwinding. Some symptoms persist because tissue still needs repair. Cough can remain after viral load falls because airways are sensitive. Fatigue can outlast fever. Muscles weakened by inactivity require rebuilding. Sleep rhythms may be disturbed. The nervous system may remain alert to internal signals. Recovery has a tail. This is why returning to normal rarely happens in one clean step. Capacity comes back unevenly. A person may feel well in the morning and exhausted in the afternoon. Appetite may return before strength. Concentration may improve before sleep. The body is not failing to decide whether it is healthy. Different systems are recovering on different schedules. The same is true after surgery. The incision may close while deeper tissue remains weak. Pain may decline while energy remains low. Blood volume, muscle mass, bowel function, inflammation, and confidence in movement may all recover at different rates. A single word, recovery, contains many clocks. Some clocks are cellular. Some are hormonal. Some are mechanical. Some are learned. Sleep interacts with nearly all of them. It is tempting to say that the body heals while we sleep, as though waking hours are for damage and sleeping hours are for repair. The truth is more interesting. Repair continues all day. Cells do not wait for darkness to clot blood or divide. But sleep changes the internal environment in ways that influence immunity, metabolism, hormones, memory, pain, and behavior. Sleep is not an empty interval. It is an active state with changing stages. During a typical night, the brain cycles through non-rapid eye movement sleep and rapid eye movement sleep. Non-REM sleep includes lighter stages and deep slow-wave sleep. REM sleep is associated with vivid dreaming, rapid eye movements, and a brain pattern that is active in a distinctive way while many skeletal muscles are temporarily inhibited. These stages repeat, but not in identical loops. Deep slow-wave sleep is usually more abundant earlier in the night. REM periods tend to lengthen toward morning. Age, illness, stress, light, temperature, substances, and sleep loss alter the pattern. The sleeping brain is not shut down. It is changing modes. Immune signals help regulate sleep, and sleep influences immune function in return. During infection, cytokines can increase the drive for sleep, especially non-REM sleep, though severe illness may also fragment it. Sleep loss can alter inflammatory signaling and reduce aspects of adaptive immune response. Studies of vaccination have shown that insufficient sleep around immunization can weaken antibody responses in some settings. The result does not mean one poor night erases immunity. It reveals that memory formation in the immune system, like memory formation in the brain, is affected by the state of the whole organism. Sleep helps coordinate timing. Certain immune cells move between blood, lymph nodes, bone marrow, and tissues according to circadian and sleep-related rhythms. Hormones that influence immunity change across the night. Adhesion molecules and signaling environments shift. The immune system does not perform the same task at the same intensity every hour. Deep sleep is associated with changes in growth hormone release. Growth hormone supports metabolism, protein synthesis, and tissue maintenance, though it is not a simple healing potion and does not act alone. Cortisol follows its own circadian pattern, generally reaching a low point near the early night and rising toward morning. These rhythms help separate and coordinate different physiological programs. The night is organized, not merely dark. Sleep also changes pain. After insufficient sleep, people often become more sensitive to painful stimuli. Brain regions involved in sensing and regulating pain respond differently. Mood and attention shift. A sore muscle that was tolerable can feel louder after a restless night. Pain, in turn, disrupts sleep. A wound throbs when position changes. A cast feels uncomfortable. Inflammation alters temperature and sensation. Worry keeps attention attached to the body. Poor sleep increases sensitivity the next day, creating a loop. Recovery sometimes depends on interrupting loops, not solving one isolated symptom. The brain uses sleep for memory and learning, which matters in rehabilitation. A person practicing a movement after stroke, learning to use a prosthesis, or rebuilding balance is asking neural circuits to retain new patterns. Sleep contributes to consolidation, the stabilization and transformation of memory after practice. Motor skills can improve between training sessions even without additional conscious rehearsal. The nervous system continues processing what was learned. This does not mean sleep guarantees improvement or that every memory is stored the same way. Different stages contribute differently to different forms of learning. Yet recovery, guided by practice, does not end when the therapy session ends. The brain keeps working on the lesson after the room becomes quiet. Sleep may also influence the movement of fluid and waste through the brain. Research in animals has described a system of exchange involving cerebrospinal fluid, interstitial fluid, perivascular spaces, and astrocytes, often called the glymphatic system. Activity appears to change with sleep and arousal state. The idea is sometimes simplified into the claim that sleep washes toxins from the brain. The evidence is more nuanced, especially in humans. Fluid movement and clearance are real topics of active research, but the exact pathways, driving forces, and importance for different diseases remain under investigation. The careful conclusion is still compelling. Sleep changes the physical environment in which brain tissue maintains itself. During waking, neurons and glia are active, ions move, neurotransmitters are released and recycled, proteins are made and broken down, and metabolism produces waste. During sleep, patterns of neural activity, blood flow, breathing, and fluid movement change. The brain's maintenance is tied to its state. The sleeping body also manages energy differently. Insulin sensitivity, appetite hormones, and glucose regulation are influenced by sleep duration and circadian timing. Repeated sleep restriction can alter metabolism and appetite, which in turn affect the resources and conditions available for recovery. Again, there is no single repair switch. Sleep is a system-level coordinator. Circadian rhythms extend beyond sleep. Nearly every cell has molecular clocks built from genes and proteins that rise and fall in roughly 24-hour cycles. The central clock in the brain responds strongly to light and helps synchronize clocks throughout the body. Feeding, activity, temperature, and hormones provide additional timing cues. Wound healing itself can vary with circadian phase. Cell migration, immune activity, and gene expression are not identical at every hour. Researchers have found time-of-day differences in some repair processes, though translating those findings into broad medical rules requires caution. The larger point is that biology has schedules. A cell does not simply ask what signal is present. It also asks when the signal arrives. Night shift work, jet lag, illness, hospital noise, and pain can disturb coordination between clocks. A person may sleep at one time while peripheral tissues remain partly aligned with another. Recovery still occurs, but the symphony loses some synchrony. This helps explain why healing can feel different in a hospital. Lights, alarms, procedures, unfamiliar sounds, and repeated interruptions fragment sleep. The environment designed for medical observation can make natural sleep difficult. Modern care increasingly recognizes that rest is not a luxury added after treatment. It is one part of treatment's biological setting. Yet sleep should not be romanticized as effortless cure. A person can sleep well and still face slow healing because of blood flow, infection, age, disease, medication, or the severity of injury. Another may recover despite imperfect sleep. Biology is resilient and multi-causal. The value of sleep lies not in perfection, but in participation. While the sleeper is unaware, immune cells change location, hormones shift, memories stabilize, pain circuits recalibrate, breathing patterns alter, and damaged tissues continue their work. The night does not heal everything. It gives many forms of healing room to continue. Every repair described so far depends on supply. Cells need oxygen to produce energy efficiently. They need amino acids to build proteins, fatty acids for membranes and signaling molecules, glucose and other fuels, vitamins and minerals for enzymes, and water for nearly every chemical reaction. Blood must carry these materials to the right place and carry waste away. This is why circulation stands at the center of healing. A healthy capillary is only one cell layer thick. Red blood cells squeeze through narrow passages and release oxygen, which diffuses into tissue. Plasma carries nutrients, hormones, antibodies, clotting proteins, and dissolved signals. The endothelial cells lining vessels do far more than form pipes. They regulate permeability, clotting, inflammation, vessel tone, and the growth of new branches. When blood flow is reduced, recovery slows. A wound at the edge of a poorly perfused tissue may remain starved despite every signal calling for repair. Cells can survive low oxygen for a time and even use low oxygen as a cue to release angiogenic factors. But prolonged deprivation limits energy, weakens defense, and can lead to cell death. The color of tissue often reveals the hidden state of circulation. Pink granulation tissue suggests active vessels. Pale tissue may be poorly supplied. Dark tissue may be dead. Clinicians learn to read these surfaces as maps of flow. Red blood cells make oxygen transport possible through hemoglobin, an iron-containing protein. Each cell is shaped like a flexible disc, maximizing surface area while allowing passage through capillaries narrower than its resting diameter. Red cells have no nucleus when mature, leaving more room for hemoglobin. They are manufactured in bone marrow under the influence of erythropoietin, a hormone produced mainly by the kidneys when oxygen delivery is low. Even the response to a local wound depends on a distant conversation among marrow, kidneys, lungs, heart, vessels, and blood. No tissue heals alone. Nutrition enters the story at the level of raw material and regulation. Collagen requires amino acids, including glycine and proline, and its maturation depends on vitamin C. Blood cell production depends on iron, folate, vitamin B12, and other nutrients. Zinc participates in numerous enzymes and cellular processes. Vitamin D and calcium influence bone. Protein supplies building blocks for muscle, immune molecules, enzymes, and matrix. But biology is not a shopping list in which more of every nutrient always creates faster healing. Deficiency can impair repair. Excess does not necessarily help and can sometimes harm. Nutrients interact, absorption varies, and disease changes requirements. The body also recycles aggressively. Amino acids are recovered from broken proteins. Iron is reclaimed from aging red blood cells. Fats are stored and released. During illness or fasting, tissues alter fuel use. The organism draws from reserves and decides which processes receive priority. This triage can be felt as weakness. During major injury or infection, metabolism may become catabolic. Stress hormones and inflammatory signals increase the breakdown of stored fuels and muscle protein. The liver changes protein production. Insulin responses shift. Even with food available, the body may process nutrients differently because it is supporting defense and repair. Recovery then includes rebuilding the reserves consumed during survival. Appetite is part of this regulation. Acute illness often suppresses hunger. In some infections, temporary appetite loss may reduce the energy spent on seeking food or change nutrient availability to microbes. But prolonged loss of intake can become harmful, especially when recovery requires protein and energy. The same response can be adaptive for a day and damaging for a month. The microbiome also participates indirectly. Trillions of microorganisms live on the skin, in the mouth, and especially in the intestine. They compete with pathogens, transform dietary compounds, produce metabolites, and influence immune development. Antibiotics, illness, diet, age, and environment change these communities. There is no single ideal microbiome shared by everyone, and the word balance can hide enormous complexity. Still, recovery from infection or medical treatment may include ecological recovery as microbial communities change again. Healing is not performed only by human cells. It occurs in a body that is also an ecosystem. Stress changes this ecosystem and the rest of repair through several pathways. A sudden threat activates the sympathetic nervous system and the hypothalamic pituitary adrenal axis. Adrenaline increases heart rate and redirects blood flow. Cortisol helps mobilize energy and modifies immune activity. These responses are useful when survival requires immediate action. A short burst of stress is not automatically harmful. It can sharpen attention, support movement, and alter inflammation in ways suited to an acute challenge. The problem arises when signals designed for brief emergencies remain elevated or repeatedly activated without recovery. Chronic psychological stress has been associated with slower wound healing and altered immune responses in a range of studies. The mechanisms may include hormones, sleep disruption, behavior, inflammation, blood flow, and changes in care. The effect is not a moral judgment. A person does not fail to heal because they worried incorrectly. Stress is physiology, context, and circumstance. This matters because illness itself creates stress. Pain interrupts sleep. Uncertainty captures attention. Loss of independence changes mood. Financial pressure, work, caregiving, and access to treatment continue even when the body needs recovery. The instruction to relax can therefore be strangely unhelpful. A nervous system does not become calm because calmness has been demanded. Safety is learned through environment, information, relationships, breathing, movement, predictability, and time. Social support can become biological support. A trusted person may help with food, transport, medication, mobility, or communication with clinicians. Their presence can reduce threat and loneliness. Touch, voice, and familiar routines can influence stress systems. Conversation can help the brain organize what happened. Humans are social organisms. Recovery evolved in groups where care from others could determine survival. This does not mean every sick person wants constant company. Immune-driven sickness behavior often includes a desire to withdraw. The useful form of support respects changing needs. closeness without demand, help without taking away agency, quiet without abandonment. Expectation also influences symptoms and recovery. The placebo effect is often misunderstood as proof that a condition was imaginary. In research, a placebo response includes changes associated with expectation, learning, context, natural fluctuation, and the meaning of treatment. The brain can alter pain, nausea, movement, stress, and other experiences through real physiological pathways. If a person expects pain relief, networks involving endogenous opioids, dopamine, attention, and prediction may reduce the experienced pain. A ritual associated with previous relief can trigger a learned response even before the active treatment takes effect. The reverse is called a nocebo effect. Expecting harm or focusing intensely on possible symptoms can increase discomfort, side effects, and threat responses. These effects do not mean belief can rebuild a broken bone without stabilization or eliminate an infection without immune control. They mean symptoms are generated by systems that integrate tissue signals with expectation and context. The brain is not a passive screen showing damage. It is an active regulator deciding how much protection is needed. This is especially important in pain, fatigue, breathlessness, dizziness, and nausea. The underlying tissue or disease matters. So do prediction, attention, memory, and perceived safety. Treatment can work at several levels at once. A reassuring explanation may reduce threat. A familiar clinician may change expectation. A visible scar may increase attention to a sensation. A frightening word may amplify it. Language enters physiology. The history of medicine contains many treatments that seemed effective because symptoms naturally improved, because attention and care helped, or because expectation changed experience. Controlled trials were developed partly to separate these influences from the specific effect of a treatment. Yet separating them experimentally does not make context unimportant clinically. A scientifically active treatment is delivered within a human encounter, and the encounter can strengthen or weaken its effect. Recovery is both molecular and meaningful. Meaning becomes particularly visible after severe illness. A body may survive an intensive care unit while the person carries weakness, sleep disturbance, memory gaps, fear, and altered identity. Muscles lost during immobility must be rebuilt. The nervous system may remain vigilant. Ordinary tasks can feel unfamiliar. Healing then is not returning to the exact person who entered the hospital. It is constructing a life that includes what happened. Scars can become part of identity. A changed gait can alter how the world is navigated. A new medication schedule can divide the day. A person may become more cautious, more grateful, more frightened, or all three. Biology does not end where biography begins. The word recovery suggests moving backward toward a previous state. Sometimes that is possible. A cut closes and the fingertip feels ordinary. A virus clears and energy returns. A fracture remodels until the bone is difficult to distinguish from its earlier form. Sometimes recovery moves forward instead. A joint is reconstructed, not restored. A brain learns a different route. A heart works around scar. A person uses an assistive device. Capacity grows in a new shape. This is not a lesser meaning of healing. It may be the deepest one. The body has always recovered by adaptation. Even a perfect-looking repair contains changed cells, new matrix, remembered immune encounters, and altered neural predictions. The organism that emerges is continuous with the one before injury, but not identical. Healing preserves identity through change. That idea is built into rehabilitation. Rehabilitation is sometimes mistaken for the final stage after medical treatment. A period of exercise is added once the real healing is finished. In fact, movement, sensation, practice, and environment become part of tissue recovery long before remodeling ends. A bone aligns its matrix with force. A tendon organizes collagen along tension. Muscle responds to load. The brain strengthens practice pathways. The cardiovascular system adapts to demand. Balance improves by being challenged within safe limits. The rehabilitation session sends information into living tissue. Mechanotransduction is the process by which cells convert mechanical forces into biochemical signals. Integrins connect extracellular matrix to the internal cytoskeleton. Ion channels respond to stretch. The nucleus changes gene expression. Cells alter growth, matrix production, and alignment. A step is not only a step. It is a message about the future forces the tissue should prepare to carry. The message must be dosed. Too much stress too early can reopen tissue or extend injury. Too little stress for too long can produce stiffness, weakness, loss of bone, shortening of connective tissue, and reduced confidence. Recovery depends on progressive challenge matched to current capacity. This is why the same instruction cannot fit every injury. Rest can protect in one phase and delay recovery in another. Movement can stimulate adaptation in one context and cause harm in another. The correct amount is a moving target. Clinicians and therapists watch pain, swelling, strength, range, control, tissue response, imaging, and function. The body reports after the session as well as during it. A useful challenge may produce temporary fatigue without prolonged worsening. A harmful one may exceed the tissue's ability to recover before the next demand. Rehabilitation is an ongoing experiment between person and biology. It is also one of the places where small improvements become meaningful. A few more degrees of joint motion can allow a shirt to be put on. A slightly faster nerve response can improve balance. One additional step can restore access to a room. Recovery is measured in molecules, but lived in tasks. Time changes every part of this experiment. A young body does not heal because it contains one universal youth molecule. It heals within tissues that generally have richer stem cell activity, more responsive blood vessels, different hormone patterns, less accumulated damage, and fewer chronic diseases. With age, each of these conditions may change. Stem cells remain in many adult tissues, but their niches alter. Satellite cells in muscle may become less responsive. Bone-forming activity can fall behind bone resorption. Blood vessels may grow less readily. Skin becomes thinner and less elastic. Immune responses can become slower in some ways and more persistently inflammatory in others. The term inflammaging describes a tendency toward chronic low-level inflammatory signaling with age. It does not mean every older person is inflamed in the same way. It describes a broad pattern influenced by senescent cells, visceral fat, microbial changes, accumulated tissue damage, and immune system remodeling. Cellular senescence is one part of the story. A senescent cell has permanently stopped dividing in response to damage or stress, but it remains metabolically active. This arrest can protect against cancer by preventing a damaged cell from multiplying. Senescent cells can also support wound healing briefly by releasing signals that shape inflammation and repair. The problem comes when they accumulate. Senescent cells can release a mixture of cytokines, growth factors, and enzymes called the senescence-associated secretory phenotype. In excess or for too long, these signals may promote chronic inflammation, disrupt nearby cells, and alter matrix. Once again, a useful temporary state becomes harmful when it does not end. Researchers are studying drugs that remove certain senescent cells or modify their signals. The field is promising but still developing, and aging cannot be reduced to one cell type. The body ages through many interacting processes: DNA damage, epigenetic change, mitochondrial alteration, protein quality control, stem cell exhaustion, immune change, and the history of every environment it has experienced. Healing in later life can be slower, but it is not absent. Older tissues still clot, inflame, build matrix, remodel bone, and learn movement. The range of possible recovery remains wide. Experience can even aid rehabilitation. An older adult may understand pacing, communicate symptoms clearly, and draw on strong routines and support. Biology changes with age, but outcomes are not written by age alone. The same caution applies to chronic disease. Diabetes can affect healing through several pathways. Elevated glucose changes proteins and vessels. Small vessel disease can reduce circulation. Immune cell function may be altered. Peripheral neuropathy can reduce pain sensation, allowing repeated injury to go unnoticed. A foot wound may therefore face poor supply, reduced warning, pressure, and infection at the same time. The wound is not failing because one cell refuses to cooperate. The entire environment has changed. Smoking can reduce oxygen delivery and alter vessel function, inflammation, fibroblast activity, and bone healing. Certain medications suppress immune or inflammatory pathways. Kidney disease, vascular disease, malnutrition, and cancer can change repair. So can repeated friction, pressure, or movement at the injured site. The local wound carries the history of the whole body. This is why two apparently similar cuts may heal differently. One closes in days. Another persists for months. Surface size is only part of the problem. Beneath it lies circulation, sensation, microbes, pressure, metabolism, and immune regulation. Modern wound care therefore asks several questions at once. Is there enough blood flow? Is infection present? Is dead tissue blocking repair? Is pressure being repeated? Is swelling limiting oxygen exchange? Is the wound too dry or too wet? Are systemic conditions being addressed? The treatment must change the environment, not merely cover the surface. The idea of a moist wound environment was a significant shift. For a long time, allowing a wound to dry and form a hard scab seemed obviously clean and protective. Research showed that epithelial cells often migrate more efficiently across a moist surface than beneath a dry crust. Modern dressings can maintain moisture while managing fluid and protecting against contamination. A good dressing is not passive fabric. Depending on design, it can absorb exudate, maintain temperature, reduce friction, support gas exchange, protect fragile tissue, and even deliver antimicrobial or biological materials. The wound becomes a controlled microenvironment. Negative pressure wound therapy uses a sealed dressing connected to gentle suction. It can remove fluid, reduce edema, draw wound edges, and influence granulation tissue in selected wounds. Hyperbaric oxygen is used in certain conditions to increase dissolved oxygen under controlled pressure. Skin substitutes and grafts provide coverage or biological scaffolding. Each method works in particular contexts. There is no universal advanced dressing, machine, or molecule that accelerates every wound. Healing resists one-size-fits-all solutions because wounds are not one disease. Fibrosis shows what happens when repair solves an immediate problem at long-term cost. After injury, fibroblasts and myofibroblasts produce matrix to restore strength. In a healthy repair, many of these cells later disappear or reduce activity, and excess matrix is remodeled. In fibrosis, matrix production continues. Collagen accumulates. Tissue becomes stiff and architecture is distorted. Fibrosis can affect the liver, lungs, kidneys, heart, skin, and other organs. The trigger may be repeated injury, autoimmune disease, infection, toxins, or unknown causes. In some tissues, fibrosis is the final common pathway of many different diseases. The scar that saves a heart after infarction becomes dangerous when similar matrix spreads through healthy muscle. The collagen that closes skin becomes deforming when excessive. Repair biology has no moral label. Its effect depends on scale and duration. Myofibroblasts are central actors. They contract and produce extracellular matrix. Their activation is influenced by transforming growth factor beta, tissue stiffness, inflammation, and mechanical tension. As matrix stiffens, cells can receive signals that encourage further matrix production, creating a self-reinforcing loop. The tissue remembers injury through mechanics. Breaking such loops is difficult. Removing the original cause may slow fibrosis, but established scar can be hard to reverse. Researchers investigate immune pathways, fibroblast states, matrix-degrading enzymes, and signals that might return activated cells toward rest. The goal is not to eliminate fibroblasts. Without them, wounds would fail. The goal is to restore an ending. Regeneration and scarring can be viewed as two answers to the same question. What should the body do when structure is lost? A scar fills the gap quickly with strong general-purpose material. Regeneration rebuilds the original arrangement of specialized parts. Scar is usually faster and safer. Regeneration is more complete, but requires precise positional information, cell sources, nerves, vessels, and developmental programs. Humans regenerate more than we often realize. Skin re-epithelializes. Bone can restore normal architecture. Liver restores mass. Blood and intestine renew continuously. Peripheral nerves can regrow. Fingertips can sometimes regenerate partially, especially in young children and when the nail-associated tissues are preserved. But humans cannot rebuild an entire limb. Salamanders can. After a salamander limb is amputated, the wound closes without forming the thick permanent scar typical of adult mammalian tissue. Cells near the injury change state. A mass of proliferating progenitor cells called a blastema forms beneath a specialized wound epidermis. The blastema is not a shapeless collection of universal stem cells. Many cells retain memory of their tissue origin and contribute to related structures. Muscle, cartilage, connective tissue, nerves, and vessels coordinate. Positional information tells cells whether they belong near the shoulder, elbow, wrist, or fingertip. The new limb grows outward in the correct sequence. Nerves are essential. Without sufficient innervation, regeneration stalls. Nerve-derived signals help maintain the blastema. The wound epidermis, immune cells, extracellular matrix, and local cells all contribute. This is not the simple regrowth of a plant cutting. It is development reawakened in an adult body. The Italian naturalist Lazzaro Spallanzani documented salamander limb regeneration in the 18th century. He observed that limbs could regrow repeatedly. Centuries later, modern imaging, genetics, and single-cell analysis are identifying the programs behind what he could only see from the outside. Why mammals scar while salamanders regenerate remains one of the central questions of regenerative biology. The difference is not that salamanders have special genes entirely absent from humans. Much of the molecular toolkit is shared. The distinction lies in how genes are regulated, how immune responses unfold, how cells change identity, and how the wound environment preserves pattern. Regeneration may be less about inventing new machinery and more about changing the sequence in which familiar machinery is used. Yet copying salamander biology into humans would require more than activating one pathway. A human limb is large, heavily loaded, and connected to a complex immune and nervous system. Uncontrolled dedifferentiation and proliferation could produce tumors or disorganized tissue. The regenerated structure would need bone, muscle, tendon, skin, vessels, and nerves aligned across long distances. Nature's example is proof of possibility, not an instruction manual. Zebrafish provide another example through the heart. After injury, their cardiomyocytes near the wound partially loosen their mature program and divide. Endothelial cells rapidly rebuild vessels. Immune cells help create a regenerative environment. Temporary matrix is later reduced. The contrast with mammalian scar formation allows scientists to compare two outcomes of a shared event. Some researchers look for factors that encourage adult mammalian cardiomyocytes to re-enter the cell cycle. Others transplant heart cells grown from pluripotent stem cells. Still others modify immune responses, matrix, metabolism, or electrical coupling. A new heart cell is not enough by itself. It must contract in synchrony, receive blood supply, connect electrically, and avoid triggering dangerous rhythms. Healing is integration. The same lesson appears in every organ. A neuron must connect to the right target. An insulin-producing cell must sense glucose and release hormone appropriately. A cartilage cell must create matrix that withstands years of compression. A skin graft must vascularize and form a barrier. Regenerative medicine tries to create those relationships deliberately. One of its most important turning points began not with a human organ, but with a frog egg. By the middle of the 20th century, many biologists believed that a mature cell became specialized by permanently losing access to most of the instructions it no longer needed. A skin cell behaved like skin, a nerve cell behaved like nerve, and a muscle cell behaved like muscle. Development seemed like a one-way narrowing of possibility. John Gordon tested that assumption by removing the nucleus from a frog egg and replacing it with a nucleus taken from a specialized cell. The egg supplied the surrounding machinery. The transplanted nucleus supplied the chromosomes. If specialization had permanently erased the instructions for building the rest of the animal, development should have stopped. Yet some reconstructed eggs developed into tadpoles, and later work showed that nuclei from differentiated cells could still support the formation of an entire organism. The experiment did not mean that every mature cell could easily become every other cell. Most transfers failed, and the egg performed an extraordinary amount of reprogramming. But the result revealed something fundamental. A specialized nucleus had not thrown away the complete genetic library. Its identity depended largely on which parts of that library were active, silent, accessible, or tightly closed. The blueprint remained. The pattern of reading had changed. More than 40 years later, Shinya Yamanaka approached the same question from another direction. Instead of placing a mature nucleus inside an egg, he asked whether a small set of genes could persuade an ordinary cell to return to a pluripotent state. The possible list was long. Embryonic stem cells express many factors that help maintain their unusual identity. Yamanaka and his colleagues tested combinations, removing candidates until only 4 were needed to reprogram mouse fibroblasts. In 2006, they reported cells that resembled embryonic stem cells in important ways. Soon afterward, human cells were reprogrammed as well. They became known as induced pluripotent stem cells. The discovery changed the emotional landscape of cell biology. Mature identity was no longer a locked room with no door. Under carefully controlled conditions, a differentiated cell could be moved backward into a state with much wider possibilities. Gordon and Yamanaka later shared the Nobel Prize for showing, across 2 very different eras of biology, that cellular specialization can be reversed. The phrase "turn back in time" is useful, but incomplete. Reprogramming does not make a cell young in every possible sense, and it does not always erase every molecular trace of its past. DNA mutations may remain. Epigenetic marks can be incompletely reset. Chromosomes can acquire abnormalities during culture. A pluripotent cell placed in the wrong environment can form unwanted tissue or tumors. The discovery opened a door. It did not remove the need for guidance. A pluripotent cell can potentially become many cell types, but potential is not function. A cell destined to become heart muscle must pass through a sequence resembling development. Different signals must arrive in the right order. Some pathways must be active for hours or days and then fall silent. Oxygen, nutrients, neighboring cells, electrical activity, and matrix stiffness all influence the result. Development is a long education. Laboratory differentiation tries to reproduce enough of its lessons. Researchers may begin with induced pluripotent cells made from a patient's skin or blood. They can guide those cells toward neurons, heart-like cells, retinal cells, or other lineages. The resulting cells can carry the patient's genetic background, allowing a disease to be studied in a dish without removing living tissue from the affected organ. This creates a quiet form of medical time travel. A mature cell is taken from the body, returned to a more flexible state, and then directed along a different developmental path. A skin cell does not simply transform straight into a neuron by wish. Its identity is dismantled and rebuilt through gene regulation. For some diseases, these cells may eventually become replacements. For many others, their first value is as models. Researchers can watch how a mutation changes development, expose the cells to possible medicines, and compare corrected and uncorrected versions. Organoids carry the idea into 3 dimensions. Under suitable conditions, stem cells can self-organize into small structures that reproduce selected features of an organ. Intestinal organoids can form crypt-like regions. Retinal organoids develop layered neural tissue. Kidney, liver, lung, and brain organoids recreate portions of development and function. An organoid is not a miniature complete human organ. It may lack mature circulation, full immune integration, nerves, mechanical forces, or the diversity of an adult tissue. Its shape may be incomplete and its development uneven. Yet it can capture enough organization to reveal how cells choose positions, communicate, and respond to disease. The remarkable part is that researchers do not place every cell by hand. They provide signals and boundaries. The cells divide, sort, fold, and specialize through local interactions. A cluster becomes patterned because each cell responds to its neighbors, the matrix, and the chemical gradients around it. That self-organization is not separate from natural healing. A wound also lacks a central foreman holding a complete blueprint. Order emerges from local rules. Platelets alter the first surface. Immune cells change the signals. Fibroblasts reshape matrix. Vessels follow gradients. Mechanical tension changes gene expression. Regenerative medicine succeeds when it gives living cells enough structure to organize themselves without allowing growth to lose direction. Tissue engineering provides scaffolds that help guide this emergence. A scaffold may be made from collagen, synthetic polymers, hydrogels, ceramics, or decellularized tissue. It can define shape, stiffness, porosity, and degradation rate. It may release growth factors or present attachment sites. Cells can be added before implantation or recruited from the body afterward. The ideal scaffold does not simply occupy space. It gives cells the right questions to answer. Is the surface soft or stiff? Which molecules can be attached to? How large are the pores? Does fluid flow through? How quickly does the material break down? Cells sense all of these features and change behavior accordingly. A bone scaffold must support load and mineralization. A nerve conduit must guide axons without blocking nutrient exchange. A skin substitute must protect the surface while encouraging vessels and epithelial closure. A heart patch must move with every beat. One material cannot imitate every tissue. Decellularization starts with a natural organ or tissue and removes its cells, leaving much of the extracellular matrix behind. The remaining scaffold preserves some of the intricate 3-dimensional architecture, including vessel pathways. Researchers can then attempt to seed it with new cells. The approach is powerful because nature has already built the geometry. The difficulty is repopulating it completely and connecting it to circulation without clotting, rejection, or cell death. A large engineered tissue faces a simple physical limit. Cells cannot survive far from oxygen. Diffusion works only across short distances. Without vessels, the center of a thick construct becomes starved. Vascularization is one of regenerative medicine's great bottlenecks. Scientists create channels, add endothelial cells, use growth factors, print branching patterns, or connect tissues to living circulation. But a functional vascular network must be leak-resistant, responsive, and connected at multiple scales from larger vessels down to capillaries. Building tissue means building flow. Three-dimensional bioprinting can place cells and biomaterials layer by layer. The printer follows a digital design, depositing bioinks through nozzles or using light to solidify patterns. The technique can create shapes difficult to make by hand and arrange several cell types with precision. Yet printing an organ shape is not the same as printing an organ. Cells must survive the process, mature, communicate, and remodel the printed material. Tiny vessel networks remain difficult. Mechanical strength and electrical integration matter. The printed structure often serves as a starting framework that biology must finish. The most important builder remains the living cell. Some of regenerative medicine's most dramatic successes began with the body's simplest urgent requirement: rebuild a barrier. In the 1970s, Howard Green and his colleagues developed methods for expanding human keratinocytes, the main cells of the epidermis, in culture. A biopsy no larger than a small piece of skin could provide cells that divided again and again, eventually forming thin sheets. The importance of that method became clear in 1984 when cultured epidermal grafts were used in the care of 2 children with extensive, life-threatening burns. Healthy skin available for ordinary grafting was limited. Cells taken from small surviving areas were expanded until they could cover a far larger surface. The sheets were delicate. They did not contain the full architecture of skin. There were no hair follicles, sweat glands, nerves, or complete dermal layers. They could tear, contract, or fail to attach. Yet they offered something essential: living epidermal cells capable of restoring a protective surface where the body had lost one. The work turned a small sample into many square centimeters of tissue. It also showed the difference between growing cells and creating a successful graft. At first, transplanted epidermis survives only if the wound bed can support it. Fluid must not collect beneath it. Infection must be controlled. The surface must remain sufficiently still. Nutrients diffuse from below until vascular connections and supporting tissue become established. The graft does not arrive as a finished replacement. It enters a negotiation with the body. Modern skin engineering tries to extend that achievement by adding dermal structure, pigment cells, immune components, vessels, follicles, and glands. Each addition makes the tissue more complete and the problem more difficult. A barrier can be rebuilt sooner than an entire skin ecosystem. Bone marrow transplantation provides another established example of rebuilding a system from its source. Blood-forming stem cells from a donor or from the patient can repopulate marrow after disease or intensive treatment. The new cells can restore blood production and, in donor transplantation, replace much of the immune system. The procedure can be life-saving, but it is not a simple refill. Before transplantation, the existing marrow and immune environment may need to be altered. Afterward, the new cells must find supportive niches, begin producing descendants, and avoid destructive conflict with the recipient. Infection, rejection, and graft versus host disease remain serious risks. The success of these treatments does not come from stem cells acting as universal repair dust. It comes from matching a particular cell population with a particular damaged system and a niche able to receive it. Gene therapy adds another route. Instead of replacing an entire tissue, it changes the instructions within cells. A functional gene may be added. A harmful gene may be silenced. Gene editing can alter a specific sequence. Some inherited blood disorders are now being treated by removing a patient's blood-forming stem cells, editing or modifying them, and returning them after preparation. The corrected cells can repopulate marrow and produce healthier descendants. This is healing at the level of information. The therapy does not repair each red blood cell individually. It changes the source so that future cells are produced differently. Gene editing also carries risks. Edits can occur in unintended places. Delivery may reach the wrong cells. Immune responses can occur. A change that is helpful in one cell type may be harmful in another. Long-term monitoring matters because the altered stem cells may persist for years. Precision in sequence must be matched by precision in context. The immune system itself can be engineered. In CAR T-cell therapy, a patient's T cells are modified to recognize a molecule on cancer cells. The cells are expanded and returned, where they can hunt targets more effectively. The response can be dramatic, but it can also produce severe inflammatory side effects and may not work against every tumor. The treatment turns immune memory and cell killing into a designed living drug. Living drugs are different from ordinary pills. They can move, multiply, change state, and persist. Their behavior depends on the patient's body. This makes them powerful and less predictable. Regenerative medicine repeatedly approaches the boundary between treatment and ecology. A transplanted cell enters a neighborhood. It meets oxygen levels, matrix, immune cells, nerves, hormones, and mechanical forces. Its fate is not determined only by what it was in the laboratory. The receiving tissue helps decide what it becomes. This is why researchers study niches so intensely. A stem cell separated from its niche may lose identity or behave unpredictably. The niche provides signals that keep it quiet, activate it after injury, orient division, and decide which descendants survive. A hair follicle stem cell, an intestinal stem cell, and a muscle satellite cell live in different niches and answer different demands. Their power comes from belonging somewhere. The dream of a universal stem cell injection ignores this belonging. Cells do not automatically travel to every damaged organ and rebuild it correctly. Some disappear. Some become trapped. Some provoke immune reactions. Some form unwanted tissue. Repair requires direction. Biomaterials, gene circuits, local growth factors, electrical stimulation, mechanical loading, and patterned scaffolds are all attempts to provide that direction. The future may involve combinations rather than one breakthrough. A scaffold supplies shape. Edited cells supply function. A drug controls inflammation. A rehabilitation program supplies mechanical signals. Sensors report progress. Imaging confirms integration. Healing has always been a network. Advanced medicine may succeed by working with that network rather than searching for a single master switch. The boundary between natural and engineered recovery will become less clear. A metal implant can already become integrated with bone. A pacemaker coordinates the heart's rhythm. A cochlear implant converts sound into electrical stimulation of the auditory nerve. An artificial joint restores movement while muscles and brain learn to use it. These devices do not regenerate missing tissue. They become partners in function. Recovery can be biological, mechanical, electronic, or all three. The comparison with other animals reveals that recovery is not one fixed ability distributed equally across life. A planarian flatworm can regenerate an entire body from a small fragment because it carries abundant pluripotent stem cells called neoblasts and positional systems capable of rebuilding missing structures. A hydra continually renews itself and can reform a complete organism from separated cells under experimental conditions. Salamanders regenerate limbs. Zebrafish regenerate heart muscle and parts of the spinal cord. Deer regrow antlers, enormous structures of bone, skin, vessels, and nerves every year. Mammalian evolution arrived at different compromises. A large warm-blooded body needs rapid closure, strong immune defense, and protection against infection. Scar formation provides speed and mechanical stability. Long-lived specialized cells preserve complex functions. Tight control of cell division limits cancer risk. The reduced regenerative capacity of adult mammals may therefore be the price of other forms of stability. This is not one proven evolutionary bargain with a single cause. It is a useful way to frame the question. Regeneration, immunity, body size, metabolism, lifespan, cancer suppression, and reproduction evolve together. A mouse can heal faster than a human partly because its body is smaller and its life moves at a different pace. A salamander's limb grows under conditions unlike those of a human adult. A zebrafish heart is small, thin, and supplied differently. Comparative biology offers clues, but every clue must be translated across scale. Even within humans, developmental stage changes healing. Early fetal skin can heal with little or no scar under certain conditions. The fetal environment contains different inflammatory responses, extracellular matrix, growth factors, and mechanical forces. As development proceeds, healing becomes more like adult repair, with stronger inflammation and scar formation. Researchers study fetal healing not to recreate pregnancy conditions in adult wounds, but to identify which parts of the response favor regeneration over fibrosis. Reduced inflammatory signaling, different collagen organization, and changes in fibroblast behavior are among the clues. Early fetal healing demonstrates that the human developmental program can sometimes rebuild skin with little or no visible scar. The adult problem is how to reopen selected parts of that program safely. The nail bed provides a small natural experiment. When the tip of a finger is lost at the right level, especially in children, some regeneration can occur. Bone, skin, nerves, and nail-related tissue may regrow. If the injury is farther from the nail-associated stem cell region, the response is more limited. Location changes the outcome because the necessary cells and signals are not evenly distributed. The body is mapped with regenerative neighborhoods. Hair follicles are another neighborhood. After superficial skin injury, cells from follicles can contribute to re-epithelialization. Sweat glands and other appendages may also provide cells. A deep burn that destroys these structures loses not only existing tissue, but local sources of renewal. The depth of a wound, therefore, changes more than the amount missing. It changes which repair systems remain available. A forest can recover differently when the soil and roots survive than when everything is removed down to rock. Healing often follows this ecological principle. Surviving structures guide what returns. The extracellular matrix stores part of the memory. It contains not only collagen but laminins, fibronectin, elastin, proteoglycans, and bound signals. Its stiffness and orientation influence cells. Damage can reveal hidden sites, release fragments, and change mechanical tension. Cells remodel the matrix, and the matrix remodels the cells. This reciprocal relationship explains why a scar can remain active long after the wound closes. A stiff matrix encourages fibroblasts to maintain tension. Altered mechanics change how cells spread and which genes they express. The physical history of injury becomes written into tissue. Massage, stretching, loading, and movement are sometimes used during scar rehabilitation, not because pressure erases collagen like smoothing clay, but because mechanical input can affect mobility, sensitivity, adhesion, and remodeling within limits. The response depends on tissue stage and individual condition. There is no single moment when healing ends. A cut may be sealed within days, but remodel for months. A fracture may allow ordinary use while internal architecture continues changing. A nerve may reconnect while sensation remains strange. A person may return to work while fatigue is still improving. Medical milestones are practical boundaries in a continuous process. Cleared for activity does not mean every cell has returned to baseline. It means the tissue and person have reached a level of risk and function considered acceptable for the next stage. The body itself uses thresholds. Enough clot to stop bleeding. Enough collagen to resist opening. Enough callus to carry increasing load. Enough immune control to reduce the response. Perfection is rarely required before progress begins. This makes recovery efficient. Systems overlap rather than waiting politely in line. Inflammation continues while new vessels grow. Matrix is produced while old matrix is removed. Movement returns while remodeling continues. Learning occurs while weakness remains. Healing is layered time. Everyday life contains many repairs too small to notice. DNA is damaged by normal metabolism, radiation, and chemical reactions, then corrected by several repair pathways. Proteins misfold and are refold or degraded. Mitochondria are divided, fused, recycled, and replaced. Cell membranes suffer tiny disruptions and reseal. Oxidized molecules are removed. Old cells undergo programmed death and are quietly engulfed. The body is not maintained by avoiding all damage. It is maintained by detecting and repairing damage faster than it accumulates. DNA repair is one of the most ancient examples. Cells can correct mismatched bases after copying, remove chemically altered bases, cut out bulky lesions, and reconnect broken strands. Different pathways handle different problems. If the damage is too severe, a cell may stop dividing, become senescent, or undergo apoptosis. Apoptosis is controlled cell death. The cell dismantles itself, packages components, and signals for removal without spilling as much inflammatory material as traumatic rupture. During development, apoptosis shapes fingers by removing tissue between them. In adult life, it removes damaged or unnecessary cells. Sometimes recovery requires letting a cell go. Autophagy is another maintenance process. Cells enclose damaged proteins or organelles and membranes and deliver them to lysosomes for breakdown and recycling. Mitophagy targets mitochondria. These systems respond to stress, nutrients, exercise, and disease. The word self-eating sounds destructive. In practice, selective destruction preserves the whole. The immune system removes cells that become infected or dangerous. Natural killer cells and cytotoxic T cells can trigger death in targets. Macrophages clear the remains. Tissue is renewed through loss as well as growth. Healing is not only addition. It is subtraction, sorting, and recycling. This becomes obvious in the transformation of a bruise. A blunt impact breaks small vessels beneath intact skin. Blood leaks into tissue. At first, the bruise may appear red or purple because hemoglobin remains within or around red cells. Macrophages gradually remove the blood. Hemoglobin is broken down through pigments including biliverdin and bilirubin, producing greenish and yellowish colors before the mark fades. The changing color is chemistry made visible. Iron is recovered and carried away. Proteins are dismantled. Fluid returns to circulation and lymph. The bruise disappears not because color is covered, but because material is processed and removed. The lymphatic system is essential to this clearance. Lymphatic capillaries collect excess fluid, proteins, immune cells, and particles from tissue. Lymph travels through vessels and lymph nodes before returning to the bloodstream. During inflammation, increased fluid leaves blood vessels, and lymphatic drainage helps prevent the tissue from remaining permanently swollen. Lymph nodes are not filters in the simple mechanical sense. They are meeting places where immune cells encounter antigens and one another. Dendritic cells carry information from tissue. B and T cells move through organized zones. A swollen node can be evidence that immune communication has intensified. The fluid around every cell is part of a moving network. Swelling after injury reflects this movement becoming temporarily unbalanced. More fluid enters tissue through permeable vessels. Proteins draw water with them. Damaged lymphatics may drain less efficiently. Muscle pumping is reduced when a limb is kept still. As vessels normalize, lymphatic flow improves, and proteins are cleared, swelling falls. In some injuries, lymphatic damage persists and chronic edema develops. Tissue then faces altered oxygen diffusion, inflammation, and infection risk. Recovery depends on drainage as well as delivery. The body continually moves between building and clearing, activation and resolution, wakefulness and sleep. This rhythm is not evidence of weakness. It is the strategy that allows a complex organism to remain stable without being static. Homeostasis is often described as keeping conditions constant. In reality, the body maintains stability through adjustment. Temperature changes across the day. Heart rate rises with movement. Blood glucose rises after food and falls between meals. Immune cells move. Hormones pulse. Health is controlled variation. Recovery is a larger excursion from that moving range, followed by a return toward workable balance. Sometimes the return is incomplete. A chronic illness creates a new baseline. A transplanted organ requires ongoing immune management. A repaired joint changes movement. A scar changes skin. The body then establishes a different form of stability. Adaptation is not surrender. It is the way living systems continue. This perspective changes the meaning of rest. Rest is not an activity at every level. While a limb is still, cells may be dividing, vessels growing, matrix reorganizing, and nerves changing sensitivity. While a person sleeps, immune cells circulate, memories consolidate, hormones pulse, and proteins turn over. Stillness at the surface can contain intense work beneath it. But rest is also specific. A healing ankle may need protection from running while the rest of the body benefits from movement. A recovering brain may tire from conversation before muscles tire from walking. A person can be physically still and mentally stressed, or physically active and emotionally calm. The body does not have one battery-marked recovery. Different systems spend and restore capacity differently. Fatigue is the felt result of several possible processes: immune signaling, sleep loss, anemia, deconditioning, pain, medication, mood, autonomic regulation, and energy metabolism. It is not laziness and not one measurable substance accumulating in blood. During recovery, fatigue can act as a brake. It reduces activity before the person consciously calculates every biological demand. Yet persistent or severe fatigue may also signal a problem requiring investigation. Protective sensations are useful, but they are not infallible. Hunger, thirst, sleepiness, pain, breathlessness, and fatigue are negotiations between body and behavior. The body sends a message. The brain interprets it. The person decides what to do within the possibilities of life. This is why recovery cannot be separated from culture and circumstance. Some people can rest. Others must work. Some have safe housing, nutritious food, therapy, transportation, and family help. Others manage wounds, illness, or disability without those supports. Biological capacity is shaped by social conditions. A technically excellent treatment can fail if it cannot be accessed, afforded, understood, or continued. A person may know that a limb needs elevation but live in a job that requires standing. A medication may work but cause effects that make daily care impossible. A rehabilitation plan may assume equipment or time that does not exist. Healing science reaches beyond the laboratory into the design of systems. Public health itself is a form of recovery. Clean water prevents repeated intestinal injury. Vaccination prevents infection. Safe roads reduce trauma. Air quality affects lungs and vessels. Workplace design prevents strain. Paid leave allows illness to run its course without forcing exposure and exhaustion. The healthiest wound is often the one that never occurs. Prevention does not make the body's repair systems less remarkable. It respects their limits. Those limits are part of tonight's quiet truth. The body is astonishingly capable, but it is not infinitely self-correcting. Some cells are lost permanently. Some scars restrict function. Some infections overwhelm defense. Some genetic changes cannot be repaired. Some pain persists. Some people follow every recommendation available to them and still recover incompletely. Healing should never be used as a promise or a judgment. The science describes capacities, probabilities, and conditions. It does not guarantee one outcome to everybody. A chronic wound is not evidence that someone failed to believe strongly enough. Long-term symptoms are not a refusal to recover. Sometimes the body has solved the most urgent problem it could solve with the circulation, energy, tissue, time, and treatment available. Survival may leave evidence: a pale line in skin, a thickened region in bone, a changed pathway in the brain, or immune cells carrying memory of an infection. These marks do not say that the injury was good. They say that something happened and the living system responded. After following the response through blood, skin, bone, muscle, nerve, sleep, rehabilitation, and regenerative medicine, the story can return to the quiet body resting here. There may be no visible wound tonight. Even so, maintenance continues. Inside bone marrow, blood cells are forming from stem and progenitor cells. Old red blood cells are being removed, their iron saved and reused. In the liver, proteins arriving from the intestine are sorted, transformed, stored, or released. Along the intestinal wall, newly produced cells are moving upward to replace those that will soon be shed. Within muscles, damaged proteins are identified and dismantled. Amino acids are reused. Mitochondria divide, fuse, and are removed when they can no longer function well. In bone, osteoclasts and osteoblasts continue their slow exchange, taking down small regions and rebuilding them according to age, hormones, and the history of mechanical load. None of this began because an emergency alarm sounded tonight. Maintenance is the older process. Healing borrows its tools and increases their urgency. The platelet that seals a vessel was already drifting through the blood. The macrophage that clears debris was already living in tissue or available to be recruited. The fibroblast that builds matrix was already maintaining the space between cells. The nervous system that learns a new movement was already changing with every ordinary experience. An injury is new. Readiness is ancient. No single cell understands the whole body. A platelet responds to an altered vessel wall. A neutrophil follows a chemical gradient. A fibroblast senses the stiffness of its surroundings. A Schwann cell changes its program beside a damaged axon. A muscle fiber responds to load. The brain compares incoming signals with memory and expectation. The coordination comes from communication rather than from one hidden commander. A molecule is released. A receptor changes shape. A gene becomes active. A vessel opens. A cell divides. Matrix tension changes. The changed matrix alters the next signal. Countless local responses overlap until a temporary structure appears. The word temporary is important. The body rarely constructs the final answer first. It builds what the next stage needs. A platelet plug slows the leak before fibrin strengthens the seal. Fibrin provides a provisional surface before new tissue fills the gap. A soft callus stabilizes a fracture before stronger woven bone and later lamellar bone take its place. A regenerating axon sends several sprouts before useful connections are selected. A person recovering movement may use an awkward strategy before coordination becomes smooth. Early repair is allowed to be imperfect because its purpose is to make later repair possible. This is one reason healing can look disorderly while it is succeeding. New vessels may grow in excess and later be pruned. Early collagen is laid down quickly and later aligned. Immune cells arrive, change state, and leave. Swelling brings fluid and proteins, but must eventually decline. The body explores, tests, and edits. The same sign can therefore mean different things at different moments. Redness near a new wound can be part of expected inflammation. Redness that expands with worsening pain and fever may signal trouble. Fatigue during acute infection may conserve behavior. Fatigue that persists can become its own medical problem. Context and direction matter. Recovery is not judged from one still image. It is watched as a sequence. Is the surface closing? Is blood flow adequate? Is inflammation resolving rather than spreading? Is strength increasing? Can the tissue tolerate a little more load without prolonged worsening? A slow movement in the right direction may matter more than one impressive day followed by collapse. Some changes are too gradual for the nervous system to celebrate. Danger demands attention immediately. Improvement often arrives by becoming less noticeable. A scar softens between 1 month and the next. A hand reaches farther without a conscious decision. A cough is absent for an entire afternoon before anyone realizes it. A hill that once required rest becomes an ordinary part of the route. One day, the body simply stops mentioning the injury so often. This may be the quietest sign of successful recovery. Attention is released from protection and returned to the wider world. Tonight, attention can also be released. The cells do not need conscious supervision. No one must direct collagen into place, remind lymphocytes what they have seen, or instruct bone where to place each mineral crystal. The body is not passive, but much of its regulation occurs below awareness. Breathing continues. Blood moves. Temperature is adjusted. Hormones change with the hour. The nervous system begins to favor the patterns of sleep. The deliberate part of the day becomes smaller while the quieter systems continue. There is no need to imagine a single switch labeled recovery turning on after the eyes close. Repair has been occurring throughout the day. Sleep changes the setting rather than performing every task itself. Movement decreases. Sensory input changes. Hormonal rhythms continue. Immune signals and brain networks enter different patterns. The organism is given another mode in which to work. Living systems are not perfectly engineered machines. They are historical, layered, and redundant. Several signals may recruit similar cells. One muscle can partly compensate for another. A neural pathway can be strengthened while a damaged one remains limited. The liver can restore lost mass. Paired organs and distributed networks provide reserve, though reserve is never unlimited. Resilience is not invulnerability. It is the existence of more than one route through change. The human body must remain open to the world. Skin must sense and exchange heat. Lungs must expose delicate surfaces to air. The intestine must admit nutrients while excluding threats. Muscles and tendons must accept force to create movement. Bones must bear load. Nerves must extend over long distances. A body protected from every possible injury would also be protected from experience. So life uses barriers that can be breached, tissues that can be strained, and systems able to respond. It does not remain unchanged. It remains continuous. That continuity is not carried by keeping every molecule forever. Most molecules are replaced. Many cells are replaced. Memories are revised. Bone is remodeled. Immune encounters alter future responses. Identity persists through organized change. Healing is one of the ways a living system changes without completely losing itself. Sometimes the result closely resembles what existed before. Bone may remodel until the fracture line becomes difficult to find. Skin may close with only a faint mark. Strength may return so fully that an old strain is rarely remembered. Sometimes recovery creates a different but workable future. A joint is reconstructed. A brain relies more heavily on another network. A person learns a new method, pace, device, or routine. Function returns in a form that could not have been predicted at the beginning. The repaired body is not an untouched body. It is a body with history. For tonight, that history can rest. Somewhere beneath the skin, matrix is being renewed. In the marrow, new blood cells are entering circulation. In muscles, proteins are turning over. In the brain, the day's experiences are being reorganized and stabilized. Fluid moves through tissues. Old cells are removed so quietly that no sensation announces their passing. No single event is dramatic. Together, they preserve continuity. The clot eventually dissolves. Extra vessels are pruned. Immune cells leave or return to surveillance. Collagen settles into a more stable pattern. The alarm lowers its voice because there is less and less need for an alarm. Recovery often reaches its destination without a final announcement. Emergency becomes maintenance again. And now the story can do the same. There is nothing more to direct tonight. The heart can keep its rhythm. The breath can arrive and leave. The surface beneath you can remain still while deeper systems continue their work at the pace living tissue requires. Healing unfolds across overlapping time. A vessel narrows in seconds. A clot forms over minutes. A surface may close over days. Strength can return over weeks. Matrix may remodel for months. A person may continue making meaning from an injury for years. Each timescale belongs to the same process. For now, only this moment is required. The room is calm. The body is not inactive, but it is allowed to be unobserved. Maintenance continues without explanation, patient and distributed, one small exchange after another. You do not need to follow it any farther. Sleep well. Good night.